Okay, good morning. Today we are going to deal with the special senses. Last time we dealt with the general senses, and today we are going to deal with special senses. Special senses will be in two parts, so today we will deal with the first part of the special senses. The special senses, as we mentioned before, include the five senses of the smell, taste, vision, hearing, and equilibrium. So today we are going to deal with smell, taste, and vision. As we mentioned previously, that the special senses are housed in complex sensory organs. They are not like the general senses, which are either in the form of, let's say, free nerve endings or just with minor modifications of connective tissue, like we have seen in the Pacinian corpuscles, the Meissner corpuscles, and other modifications for general sensations. Here, as you will see, that it is more complex sensory organs. And some of these sensations are like the olfaction and gustatory sense that we are going to deal with today. These receptors are chemical receptors. Others, for vision, they are photoreceptors. So we will start with the olfaction. And the olfactory epithelium is, in fact, is located in the upper part of the nasal cavity. Probably we mentioned that previously, that most of the mucosa of the nose is lined with respiratory epithelium. Remember the pseudostratified columnar ciliated epithelium with goblet cells. This is the typical respiratory epithelium, but the roof of the nose, including the upper part of the nasal septum, is lined with olfactory epithelium that contains the olfactory sensory cells. And this roof of the nose is made by the cribriform plate of the ethmoid bone. If you remember the lecture on the skull, that in the anterior cranial fossa, there was a cribriform plate. Cribriform means sieve-like. And so it's a cribriform because it has a lot of perforations, and these perforations are produced by the fibers of the olfactory nerves that pass from the olfactory mucosa to the anterior cranial fossa. It is part of the ethmoid bone of the skull. So these are the, this is how the olfactory epithelium is organized. We have olfactory receptor cells. These are chemoreceptors. They are more or less, they are neurons, the type of bipolar neurons. Remember we mentioned that neurons could be multipolar neurons, bipolar neurons, or pseudo-unipolar neurons. This is a bipolar neuron. It has a process that represents a dendrite, and the other process is the axon. And these axons of these olfactory receptor cells, they pass through the cribriform plate of the ethmoid bone to reach the anterior cranial fossa. The dendrite, as you can see here, the dendrite, or the process that represents the dendrite, has multiple cilia, but these cilia are non-motile. On the other hand, they contain the olfactory receptors. So the olfactory receptors are present on the cilia of the dendrite. And in between these olfactory receptor cells, there are supporting cells. So the supporting cells, they act as insulators, they support the olfactory epithelium, and they are, as you can see, they are columnar cells. And in addition, there's a third type of cell, these blue-colored cells. They are short cells located at the base, so they are called the basal cells. And these cells are actually, they are the stem cells, the cells that can multiply and produce more of these olfactory receptor cells. This is rather something unusual about the nervous system. As we mentioned previously, that the nervous system, the neurons, they do not replicate, they are not replaced, but this is probably the only exception here, that these cells can replace the olfactory receptor cells, can multiply and replace the olfactory receptor cells, and this happens once in a month. So three types of cells are present in this olfactory epithelium: the basal cells, which are the stem cells; the receptor cells, olfactory receptor cells, which have a dendrite that has non-motile cilia containing the receptors; and in between them there are cells that are supporting cells. And in addition to that, we need some mucus secretion. So this mucus secretion here is provided by glands. You can see here, these are exocrine glands. So these are olfactory glands. They have a duct here and they secrete mucus. So it is unlike the respiratory epithelium where you have goblet cells, but here you have glands and they secrete mucus, and this mucus will act as a medium for the material to be smelled, to dissolve in it. So that's why the sensation here, or the receptors, are chemical receptors. They need the material to be dissolved in the mucus. So that is how the olfactory epithelium looks like. Then, as these axons, they pass through the cribriform plate of the ethmoid bone, they are going to synapse in a place here in the anterior cranial fossa, which is called the olfactory bulb, immediately above the cribriform plate of the ethmoid bone. You can see it here. This is the olfactory bulb. These are the axons passing through the cribriform plate. And then from the olfactory bulb, the axons, or second-order neurons, they will reach the olfactory cortex. In fact, the olfactory cortex is mainly present here in the temporal lobe, and most of these fibers, they don't need to pass to the thalamus. The thalamus, although it's a hub for all types of sensation except olfactory sensation, the fibers can reach the cortex without passing through the thalamus. Yet some fibers, they go to the thalamus. Other fibers, they go through the thalamus, they go to the limbic system, and this is the basis why there is some sort of emotional response to odors. Some odors might bring back memories because of the involvement of the limbic system. Remember, the limbic system, or the limbic lobe that we mentioned about, is related to not only for olfaction, but it is for memory, for emotions, anger. So that's why emotional responses are combined with odors, and memories are also combined with odors because of the involvement of the limbic system. So this is all about olfaction. Then I will deal with the second sensation here, the special sense, gustatory sense, or taste. Again, taste requires the dissolving of substances, and this dissolving of substances here is taking place because of the presence of the saliva in the mouth. So you don't need like other mucus secreting glands like what we have in the olfactory epithelium, and these are the 5 classes of stimuli: the sour, the bitter, the sweet, salty, and the new one that's called umami. I don't know if you have heard of this, the umami smell, the umami taste, which is the meaty taste, like the taste of meat or tomato.
I'm sure you're kind of going, wait a minute, my food wouldn't taste that great. Exactly. Because we all grew up learning that there are 4 basic tastes in our mouth, right? We all know it's sweet, sour, salty, and bitter. And we believed that for a long time until along comes this thing called umami. And what we discovered is that umami is indeed a 5th taste, which means that inside our mouth, we have the ability to perceive the 5 different tastes. And that's kind of important because each taste has a reason for being there. So for instance, if I'm eating something sweet, what that's telling my body is it's all about calories and it's about consuming energy. Or if I'm, let's say, eating something bitter, well, what that's transmitting to my brain is that be careful, maybe that's a poison. But through social training, we actually learn to like things that are bitter. What about umami? Umami is made up of that glutamic acid, an amino acid that's one of the building blocks of protein, which means that umami is really our body's way of perceiving or tasting protein. So when we eat something that has umami in it, not only is the message going to our brain that we're consuming protein, but it's also giving this message that, wow, this is really good. Now, interestingly enough, I haven't always believed in umami. In fact, I, like a lot of people, were sort of on the fence if it really existed, and certainly I didn't want to use MSG. But one day, in one of my classes I was teaching, I remember talking with a student from the Philippines, and in the lecture that day I mentioned about how we don't use MSG in our kitchen because it's a little suspect or whatever. And afterwards, the student came up to me, and she said, you know, Chef, I don't get it. What is it about MSG? Because when I'm at home, my mom has it in the kitchen, and we grew up eating it on a lot of things. Now all of a sudden, I've sort of got this question mark in my head. And then she says, Chef, have you ever tried it on scrambled eggs? And so right then in the kitchen, we made some scrambled eggs, because I was curious. And she put in the MSG, and I couldn't believe the flavor boost. And it was that day that I started thinking, you know, maybe there's something to this umami thing.
So maybe there's something that is called umami, but it is there. And so here, the taste buds, they are present on the tongue. The taste buds are present on the tongue. They are also present on the soft palate, which is behind the hard palate, and they are present on the epiglottis. Remember this leaf-like cartilage, which is covered by mucous membrane that covers the inlet of the larynx, which is called the epiglottis. So it is all these places, they contain taste buds, and it is found on papillae. There are different types of papillae on your tongue. You can see that in the mirror. So most of these papillae that you see them in the mirror are actually filiform papillae. They give the tongue this velvety appearance or velvety texture. But these, the filiform papillae, which are mostly occupying the surface of the tongue, they do not contain the taste buds. The taste buds are present in other papillae. These are called the fungiform papillae. You can see them. They are flat-topped. You can see them on your tongue. And also they are present in other papillae on the side of the tongue, which are called foliate papillae. And also there are other papillae which are called vallate papillae or circumvallate papillae, which are located most posteriorly. It will be very difficult for you actually to see that if you pull your tongue out. These will be very difficult to see. But you can clearly see the fungiform papillae in between the most prominent filiform papillae. Remember that these filiform papillae, they do not contain the taste buds. Taste buds are present in other type of papillae. So a taste bud will look like this under the microscope. It's an oval body. Again, there are three types of cells. So there are supporting cells and there are the receptor cells, similar to what we have in the olfactory epithelium, and there are basal cells, which are the stem cells. So these supporting cells, actually, they are columnar cells, and they surround a bunch of gustatory cells or receptor cells, about 50 of them. And these receptor cells, they have microvilli, and the microvilli, they will appear on the surface of the taste bud through an opening, which is called a taste pore. And these cells, actually, they are modified epithelial cells, and so they can be replaced. The supporting cells can replace them. In fact, the basal cells are continuously multiplying, replacing the supporting cells, and then the supporting cells will differentiate into gustatory receptor cells. And this process takes place in about like 7 to 10 days. So that's why when some of these receptors are destroyed, because if you drink a very hot coffee, then you can regain the taste after a few days, because these are replaced, always they are replaced by the basal cells forming the supporting cells, and then the supporting cells will form the gustatory receptor cells. And then actually it is the sense of smell, the sense of taste, they are so closely related together, and probably you have discovered that when you have a cold, or if you have allergy and your nose is blocked, then you will not feel the taste of food, because most of our feeling of taste of food is actually related to the smell. So if your nose is blocked, then you will not taste much of the food. The gustatory pathway will start from nerves, or the dendrites of nerves that will take the impulses or sensations of taste from the tongue. We have 3 of the cranial nerves are involved. In the anterior 2/3 of the tongue, this is the part of the tongue that we always see when we pull our tongue out. The anterior 2/3 of the tongue, it is the facial nerve, the 7th cranial nerve. And then in the posterior third of the tongue, it is the glossopharyngeal nerve, the 9th cranial nerve. Remember, cranial nerve are 12 pairs of cranial nerves, and we usually use Roman numerals to identify them. So the facial is number 7, and the glossopharyngeal is number 9. And then the soft palate and the epiglottis, those taste buds that are present there, the impulses are carried by the vagus nerve. All these 3 cranial nerves, number 10, 9, and 7, they carry these taste impulses to the medulla oblongata, where there is second-order neurons will be projected from the medulla. There's a gustatory nucleus here, and this will be projected to the thalamus. See here, this sensation has to pass through the thalamus. So, and then third-order neurons are projected to the sensory cortex of the cerebral hemisphere. So here again we have 3-order neurons. Remember the sensory pathway, the general sensory pathway, where you have 3-order neurons. So and then the sensory area is in the parietal lobe, and part of the gustatory area is also located in the insula, the part of the cortex that is hidden from the remaining part of the cerebrum. The last sensation we are going to deal with today is the visual sensation, and these are photoreceptors that are present in the retina of the eye. But before dealing with them, I would like to talk about the anatomy of accessory structures of the eye, and these include the eyelids, eyelashes and eyebrows, and the lacrimal glands. So the eyelids here, the Latin name is the palpebrae or palpebra. So the space in between the 2 eyelids is called the palpebral fissure. So this is the palpebral fissure, the gap between the eyelids. There's a muscle that elevates the upper eyelid, which is called levator palpebrae superioris. Palpebrae means an eyelid, okay? And then these eyelids, they protect, of course, they protect the eye, and they lubricate the eye. There are sebaceous glands. Some of these sebaceous glands are related to the eyelashes, to the hair follicle of the eyelashes, and when these sebaceous glands get infected, you will have something like the boil on your skin, but here it is called a style. But there are other sebaceous glands which are present in the eyelid, separate from the eyelashes. They call them the meibomian glands. You don't need to worry about this name, but just to tell you that there are multiple sebaceous glands here, and the importance for these sebaceous glands is that they lubricate. In fact, it is not only the sebaceous gland that is lubricating the eyeball, but it is the tears that are produced by the lacrimal gland. And so the thin film of tears, the water, is spread by sebum, which is a little bit oily material, and this will reduce the evaporation of water. So you don't need to produce a lot of tears because it is already will be covered by a very thin layer of sebum from the sebaceous glands that are present in the eyelids. And then we have the eyelashes and the eyebrows, which protect from foreign objects, from perspiration, and from sunlight. So there's a lot of protection here to the eyeball. In addition to that, there is the lacrimal apparatus. And this lacrimal apparatus consists of a lacrimal gland. Look at the location of the lacrimal gland. It is located in the superolateral part of the orbit. And part of it extends to the upper lid. But mainly it is present in the orbit. The upper lateral part of the orbit, you have this gland. This is an exocrine gland. So when it secretes, it will secrete through ducts, and these ducts will open into the region here, which is called the conjunctiva, the mucous membrane that covers the eyeball anteriorly and lines the eyelids. It's called the conjunctiva. So these ducts, they open into the conjunctiva, and they will wash the front of the eyeball. And as you are blinking, then this fluid is going to spread in front of the eyeball with blinking, and then the fluid is drained to the medial side of the eye. This region is called the medial angle or the medial canthus of the eye. If you notice that, or if you remember that there was a muscle on the face which is called orbicularis oculi, which causes closure of the eyelids and is responsible for blinking, orbicularis oculi. If you put your finger on the lateral angle of the eye and blink, you will notice that there is that the tip of your finger is pulled medially, because as you are blinking, it's not only closing the eyelids, but there is a medial movement of the eyelids, and this will always wash or sweep the fluid coming from the lacrimal gland toward the medial side. If you put your finger here on the lateral side and blink, you will see that your finger is moving a little bit medially because of this medial movement that causes the tears to go to the medial side. And here there is an opening on either of the eyelids, which is called the lacrimal punctum. You can see that if you roll down your eyelids in front of the mirror, you will see that the medial side of the eyelid is hairless, and there's a small, tiny black spot there. This is the opening, the lacrimal punctum, and this will drain the excess tears from the conjunctiva to a lacrimal sac, and then from the lacrimal sac there is a duct which is called the nasolacrimal duct, which will take it down to the nose. Okay, so that's why excessive tears, they will go to the nose. If there's too much tears, they will spill on the cheek. But and also when you cry, you start blinking a lot in order to try to wash out the excessive tears, and then these excessive tears will reach the nose, and that's why when you cry you sniff, because there's excessive tears reaching the nose. That's how the lacrimal apparatus works. There is a lacrimal gland, and then there are puncta, lacrimal puncta, lacrimal sac, and a nasolacrimal duct. About 1 ml of tears are produced a day, not that much. And as I said, they spread over the eye by blinking. They are important for preventing dehydration. They have bactericidal enzyme, and they are also important for the nourishment of the cornea, as we will see in a moment. Inside the orbit, there is the eyeball. So this is the eyeball here, And we will see that the eyeball has 3 coats, 3 coverings, but the eyeball is moving as we are. We can move the eyeball because of the presence of muscles. The muscles that move the eyeball are called extraocular muscles because they are present outside the oculus. The oculus is the eye. So they are present outside the oculus, so they are extraocular muscles. They are called extraocular because there are other muscles which are present inside the eyeball. So I'm now talking about the extraocular muscles, and these are skeletal muscles. It's our under control skeletal muscles. There are 6 of these muscles. 4 of them are called recti, rectus muscle. So we have, like, for example, superior rectus, inferior, medial, and lateral rectus, 4 of them. Rectus means straight, and 2 are oblique. So there are, in total, 6 muscles, and they are supplied by the 3rd, 4th, and 6th cranial nerves. So here the cranial nerves supply these muscles. So now you know that the first cranial nerve is the olfactory. The second cranial nerve I'm going to talk about in a moment is the optic. The 3rd, 4th, and 6th are concerned with the control of these extraocular muscle. And the 5th cranial nerve is in fact the trigeminal nerve, the nerve that is sensory for the face and motor for the muscles of mastication. The 7th cranial nerve I have just mentioned, it is involved as one of its duties is that it carries taste sensations from the anterior 2/3 of the tongue. The layers or the tunics of the eyeball, they are fibrous coat, the outer layer, vascular layer, and the nervous layer, the inner layer. In fact, if you look closely at these layers, you can compare them with the brain and the meninges. So the outer layer is the hard, tough layer, which is called the fibrous tunic, which consists of the cornea and the sclera. It is equivalent to the dura mater of the brain, the hard covering of the hard meninges of the brain. So the fibrous layer partly is the cornea, which is located anteriorly, and the other part is the sclera. The sclera is the white of the eye. The cornea, the sclera is the white of the eye, and the cornea is the transparent part which is located anteriorly. It is transparent because it is non-vascular. So that's because it depends on its nourishment on the tears, on the film of fluid. It depends on its nourishment on from the blood vessels that are present in the nearby white area, the sclera, and it depends on its nourishment on a fluid that is located here, what we call the anterior chamber of the eye. I'm going to talk about this fluid in a moment. So it is non-vascular, but still it can receive nutrients and oxygen from the nearby structures. This is the major refractory part of the eye. So it causes refraction, in addition, of course, to the lens, but mainly it is the cornea. The lens of the eye is for the fine adjustment of refraction. The reason that it is the reason that it's transparent is not only that it is avascular, but because the fibers here, the connective tissue fibers, the collagen fibers, they are parallel fibers. So they are not in different directions. So they are parallel fibers, and this physically results in the fact that they become glassy and transparent. Parallel collagen fibers. That's why when this cornea is injured, like for example corneal ulcer, then it is going to be replaced by scar tissue, and this contains irregular collagen fibers, and this results in opacities of the cornea. Okay, because it is avascular, then it can be easily transplanted, and the transplantation of the cornea are common and successful. So no blood vessels means no antibodies to cause rejection. It is nourished by tears, as I mentioned, and by the aqueous humor. The aqueous humor is the fluid that is present here in the anterior chamber of the eye. I'm going to talk about it in a moment. The second part of the fibrous layer is the sclera, which is the white of the eye. And this consists again of dense irregular connective tissue, collagen fibers, fibroblasts, but it is not transparent and it is vascular. In fact, its vessels help in the nourishment of the nearby cornea. It is very hard. It provides the shape and support for the eye, for the eyeball. And these muscles, the extraocular muscles, are in fact attached to the sclera because it is hard. So it provides shape and support. And as you can see here, at the junction of the sclera and the cornea, this one, there's a venous sinus, circular shaped venous sinus. I'm going to talk about it in a moment. It is called the scleral venous sinus, or they call it the canal of Schlemm. Posteriorly, the sclera is perforated by the optic nerve, the second cranial nerve. So again, to remind you that I'm talking about the layers of tunics or layers of the eyeball, 3 layers, a fibrous layer, which is the counterpart of the dura mater, and this consists of a transparent cornea and the white, hard sclera. Now we're going to talk about the vascular layer, the vascular tunic, and this is the counterpart of the arachnoid and the pia mater of the meninges. So it consists of the choroid. This is the part located posteriorly. About the posterior 4/5 of it is called the choroid, and the choroid contains pigmented cells, melanocytes. So it's black in color and it's very highly vascular. It's black in color, and the reason for that, the black pigment, is that to absorb the scattered light. So the light will not be scattered when it falls on the retina. It will not be scattered here and there and results in a blurring of vision. And it is highly vascular, and so it provides the blood source for the inner layer, which is the neural layer. Because it is highly vascular, sometimes when you have photographs, when you take photographs with a flash, you will find that there's a red eye reflex through the pupil, because the eye is reflected through the vascular choroid. It's highly vascular. Okay? Anteriorly, so posteriorly it is the choroid, that's the vascular tunic. Posteriorly it's the choroid. Anteriorly it becomes the ciliary body, and further anteriorly it is the iris. So the ciliary body, as you can see it here, this is a close view of the ciliary body. It's like a ring around the eye, but we are looking at it here in section. So it is a distended part, and it has folds, or what we call processes. So there are ciliary processes here. These processes, they secrete fluid. This is the aqueous humor that I mentioned about. Again, I'm going to repeat that in a moment. So they secrete fluid. Look at the arrows here. This is the direction of the fluid that is secreted by these ciliary processes. So there is fluid going from the posterior chamber to the anterior chamber, and then it is drained by this venous sinus, the canal of Schlemm, which is present at the junction of the cornea and the sclera. And inside the ciliary body, there are smooth muscle fibers. Okay, so these are smooth muscle fibers here, and attached to the ciliary body, there are ligaments, which are called suspensory ligaments of the lens, because they suspend the lens. And when these ligaments get tightened because of contraction of ciliary muscle, the lens will become flat. And this is how, if they are relaxed, then the lens becomes fatter, becomes more concave, and if they are controlled, if the muscle fibers are contracted, they will cause tension of the suspensory ligaments, and the lens will become more flat. And this is how the fine adjustment of reflection or refraction of light will take place. Most of the refraction is taking place at the cornea, but here this is the fine adjustment, and this is helped by the contraction of these muscles, that smooth muscle fibers that are located in the ciliary body. So the ciliary body, it controls the concavity of the lens, and it is important for the secretion of the aqueous humor here by the ciliary processes. And then we have the pupil, the iris. The most anterior part is the iris, and the iris has an opening which is called the pupil. And you can see here the comparison between the arrangement of the iris and pupil and the arrangement of the camera. Here you have the diaphragm is the iris, and the aperture is the pupil. So you can change the opening of the aperture, and here you can change the also you can change the size of the pupil. Of course, this takes place involuntarily because the pupil contains, again, it contains the smooth muscle fibers. So these muscle fibers are in two arrangements. Either they are called dilator pupillae, and these are radially arranged. So when they contract, they will open, they will increase the aperture because they are radially arranged. So dilator pupillae, or the other group of these muscles, they are smooth muscle fibers again, they are circularly arranged. So when they contract, they will cause the pupil to become narrower. Okay? So we have dilator pupillae and sphincter pupillae. Definitely you have noticed that your pupil changes in size in dark and in light. So when there is when light is projected on the eye, you will immediately see that the pupil will become narrowed because of the contraction of the sphincter pupillae muscle. And the sphincter pupillae muscles, both of them, the dilator and sphincter, are supplied from the autonomic nervous system. The dilator pupillae is supplied by sympathetic fibers, while the sphincter pupillae is supplied by parasympathetic fibers. Bless you. So, dilator pupillae, again, supplied by sympathetic fibers. Okay? Remember that when you are stressed, when you are angry, your eyes are wide open and your pupil will become dilated. And the parasympathetic fibers will cause narrowing of the pupil because of the contraction of the sphincter pupillae muscle. So there are muscle fibers inside the eyeball, either in the ciliary body or in the iris. But these are smooth muscle fibers, unlike the skeletal muscle fibers which are present, which rotate the eyeball as a whole. Iris recognition nowadays is used as a method of biometric authentication because in the iris there are a lot of folds, ridges, and crypts. So, and again, the iris contains melanocytes and melanin pigments, and this is the reason that the colors of the iris are different depending on the amount and type of melanin that is present in these melanocytes. So the iris is gives the color of the eye, and the dark area here is called the pupil. It is the opening here, and of course all of this is covered by the cornea. We cannot see because it is transparent, and the white of the eye is the sclera. Okay, and the opening is the palpebral fissure. The lens is biconcave. It is, again, it's avascular, so it's transparent, and it contains fibers which contain a protein called crystalline, and it is clear, perfect, completely transparent. There are no blood vessels in it, and it is held in place by the suspensory ligaments that are connected to the ciliary body. And as I said, it focuses the light on the retina, fine adjustment of focusing the light on the retina. With age, with certain diseases, this normally transparent retina will become a little bit opaque, and this is what we call cataract. So cataract is, as you can see here, that the lens, you can see the lens, the opaque lens, through the pupil. This is what you see here, and this is the view that this patient can see. It looks like there's a fog on the picture, because the opacity of the lens will be, the lens will become opaque and it's no more transparent. This is what we call cataract, and it can be, the lens can be replaced. The deeper layer is the nervous tissue, so it represents the brain. Remember, as I said, that there are 3 layers. So there's the outer layer, the fibrous layer, and then the vascular layer, and then the deeper layer, which is the nervous layer. And this is the retina, and you can see it is located in the posterior 3/4 of the eyeball. This is the deepest layer, and there is a part here in the retina which is called the macula, or the macula lutea, and just lateral to it is an area which is called the optic disc. This is where the optic nerve passes out of the eyeball. So this is called the optic disc, or they call it the blind spot as well. This is how you look at the inside of the eyeball. We're using the ophthalmoscope, a special instrument which is called the ophthalmoscope, and this is the look, how it looks inside the retina. You can see here, this is the region of the optic disc. This is a blind spot. There are no receptor cells here at all, and just lateral to it is the region of the macula lutea, which contains most of the sensitive or photoreceptor cells are located here, and you can see that there are blood vessels as well in the view. So this is the retina. The retina is in multiple layers. Generally, it has a pigment layer. This is a pigmented layer, and this pigmented layer has the same function as the choroid. Of course, here, the choroid should be located here in this location, and again, it will, the pigmented layer doesn't contain neurons. It only contains the connective tissue, melanocytes, and absorb the excessive light. And then we have the neural layer. The neural layer, we have multiple cells. Some of them are used for multiple neurons. Some of them, they modify the sensations, but the most important of these neurons are the rods and cones. The shape according to their shape. So they are either rod-like or they have the shape of a cone. That's why they are called rods and cones. And these are the photoreceptive cells, the rods and cones. And once they receive the sensation, they will transmit it to the other layer, which is called the bipolar neurons, and then to the ganglion cells, ganglion layer. So these ganglion cells, they they have a peripheral process. These peripheral processes together, they will constitute the optic nerve. So all these processes arising from the ganglion cells, they will accumulate here at the optic disc and leave the eyeball through the optic nerve. So the region of the optic disc is only fibers, it's only axons. There are no neurons, photoreceptor neurons. They are not present there. That's why it's called the blind spot, because all these axons accumulate there and leave the eyeball at one location. The rods, they are more numerous than the cones, as you can see here. They are more numerous. They are distributed everywhere in the retina, but the cones are less numerous, and they are very important for color vision. While the rods, they are important for night vision and for shades, and they are present everywhere. Not all the animals, they have cones. Like, for example, dogs, they only have rods, they don't have cones, so they are colorblind. And also, in people who are colorblind, they have some problems with their cones, because these cones, actually, there are 3 types of cones perceiving different colors. Some of them are for the red, others are for the green, and others for the blue. So the red, green, and blue. Okay? And as I said, they are mainly concentrated in the region of the macula lutea. This region here, in the macula lutea, which contains a depression called fovea centralis. It's a very tiny area, but is very important for the acuity of the vision and for the color vision. Otherwise, most of the retina contains the rods, a little bit of cones. Regarding the cavities here of the eyeball, we have an anterior cavity, anterior space, and a posterior space. The anterior space contains a fluid, the aqueous humor, which I just mentioned about. It is produced by the ciliary processes. And the posterior space here, the posterior cavity, contains a gel-like material, which is important for the shape or the shape of the eyeball, and it is jelly-like. It's called the vitreous body or vitreous humor. But the aqueous humor is constantly being produced and drained, something like the cerebrospinal fluid, which has a circulation. The vitreous body or the vitreous humor is produced during embryonic life, and it is still there. It doesn't have circulation, and it is not renewed. So the aqueous humor is produced again by the ciliary body, by the ciliary processes, passes through the posterior chamber of the eye, which is located between the iris and lens, and then goes through the pupil to the anterior chamber of the eye, which is located between the iris and the cornea. And here at this angle here, which is the angle between the cornea and the iris, iridocorneal angle they call it, there is the canal of Schlemm, the venous sinus, and it drains it back to the circulation, to the blood. If this angle is closed, there's a problem here in the drainage of this fluid, of the aqueous humor, then this will result in accumulation of the aqueous humor inside the eyeball, increase the pressure inside the eyeball, and result in a condition which is called glaucoma. So we have glaucoma, which is increased in the pressure of the eyeball, and the cataract, which is the opacity of the lens. As the impulses leave the retina, they will leave it through the optic nerve. The optic nerve has a partial crossing or decussation at a place which is called the optic chiasma, and it will form the optic tract. The optic tract reaches this area, this nucleus, which is in fact located in the thalamus. Again, the thalamus is involved here, and the nucleus is called the lateral geniculate nucleus. Lateral geniculate nucleus is part of the thalamus. So this pathway, sensory pathway, again passes through the thalamus, and then from the thalamus, the fibers will project through what we call optic radiation to the cortex. The cortex is the visual cortex, and it is located in the occipital lobe of the brain. So from the retina, through the optic nerve, optic chiasma, optic tract, to the thalamus, optic radiation, to the primary visual area, which is located in the occipital cortex of the brain. Yes?
What about something called dry eyes? What causes that?
Well, the dry eyes is something that is related to the lacrimal apparatus. It is not related. These fluids are inside the eyeball. The dryness which you feel is outside the eyeball. Yeah. It's not related, no, not to these humors. Yeah. Any other question?